Hello, my name is Emily Benjamin, and I'm a marine scientist working on restoring Greenland mussels to Pelorus Sound in New Zealand. And this video is going to take you through some of the work that we've been doing on this 3-year project. So this project is a collaboration between many organizations, primarily of which is the Marine Farming Association. And that is an association made up of different marine farming companies. And some of the marine farmers got together to design this restoration project in the very beginning, more than 3 years ago. And what they wanted to do was to restore Greenland mussels back to the seabed where they used to thrive. And then got involved with the University of Auckland, NIWA, and the Nature Conservancy to design this restoration project. So why restore Greenland mussels to the seabed when they're already growing on lines through aquaculture? Well, just like they do on aquaculture lines, on the seabed, they also build habitats, and they help to bind sediment and create habitats for other animals. And these include lots of juvenile fish and different invertebrates like starfish and sea cucumbers. And they do this on the aquaculture lines, but when they're on the seabed, they can produce these types of habitats and build these reefs that really help stabilize sediment and build habitats for creatures that live close to the seabed. They're also incredible water filters. So a single Greenland mussel can filter up to a bathtub of water in a day. And they're a great economic resource for New Zealand. As I said, they grow on lines for aquaculture. So we have an available source of mussels from these lines to put on the seabed to try to restore these benthic environments. So a little bit about the decline of wild mussel populations in Pelorus Sound. So here we have a dredging boat where they used to take these boats and kind of rake the seabed to collect Greenland mussels through different parts of New Zealand. But the part we're going to focus on today is Pelorus Sound. And this is a map of Pelorus Sound where you can see that there is different types of mussel populations on the seabed. So this map is from the late 1960s, and the green here is Greenland mussels. And you can see in the late 1960s, there used to be thriving Greenland mussel populations on the seabed. But after this time, they continued to be dredged and handpicked from the shoreline. And what happened was these mussels became completely wiped out from the seafloor. And we don't see any natural mussel populations occurring in these places where they used to today. Even though there is mussels growing on lines, creating larval supply to this area, we aren't seeing any natural recovery. And that's why we wanted to take the time to intervene and try to restore these mussel beds and see if we can initiate some natural recovery processes in this area. So just to clarify exactly where we're looking here. So here we have a map of New Zealand, and zooming into the top of the South Island in the Marlborough Sounds is Pelorus Sound. And this is where we're looking to restore these mussels based on these maps that we have from the late 1960s of where mussels used to live. And so we initially started out to understand if we can take mussels growing from aquacultured lines and put them on the seabed, and if they can still survive on the seabed in places where they used to, even though the seabed has seen a lot of changes since then. So the seabed, without the mussels there, has sort of a really muddy environments and things like that. So we wanted to see, can mussels survive? So our very first effort more than 2 years ago was to put these aquaculture mussels in 5 locations throughout the inner Pelorus Sound. And this is a map here showing you those 5 locations. So we scattered these locations throughout to test these different areas and see if mussels can survive there. So working with the marine farmers, what we did was we collected mussels from these growing lines, which you see here. And mussels grow on these lines in clumps, and they sort of form these natural clumps with their byssal threads, also known as their beards, and they attach to each other, which builds stability for them. So we wanted to keep them in those clumps. So one of the marine farmers created an idea to sort of strip the lines in a very specific way to try to keep the mussels clumped together. And then we filled these harvesting bags that they typically use for harvesting. And then a marine farming barge, the company Aroma, dropped these bags down, and NIWA scientists, including myself, went down and cut these bags open and sort of put these mussels into a square plot, which you can see here. And these mussels, we did this to create little plots, 3 little plots in each of those 5 locations originally. And this is what those restoration sites look like. So here we have, in this light blue circles in the middle, these pie charts, we have sediment composition. So what we did was we went down and we collected sediment and tested it to understand what kind of sediment is at each of these locations. So here we have, most of these lighter colors are sand, and then the dark blue color here is mud. And so what we wanted to do was test a variety of sediment composition locations. So that's how we picked those 5 locations originally. So we have that outer site in mid-Pelorus called Grant Bay. And as you can see here, it's very rocky, and there's low mud content, low dark blue color here. And then going down the Hickapoo Reach towards Pelorus Sound, we have Maori Bay, which is a mixture of sand mostly, but has a little bit of mud. And then we move in Pelorus Sound, where we used to see those historical beds. And we have 3 locations there, but I'm only showing 2 here. And what these are is, these are kind of a mixture between sand and mud. And then we have 2 sites that are more than 50% mud content. So we wanted to see if the mud content would affect the mussel survival. And this is what the mussels looked like in these plots after 5 months on the seabed. So you can see here already the mussels had become established, and they were already building habitats. You can spot some different organisms here, including starfish and sea cucumbers. And it didn't seem in the beginning that this mud content was affecting the mussels in any way. So how we monitor these mussels is we drop down via scuba, and we are measuring how the mussels are growing. We're measuring how they're surviving. We're looking for predation. So are they being, what's eating them? Is it starfish or regular fish or a variety of organisms? We're looking for how the mussel bed itself is changing, and things like that. So we monitor these mussel beds many times throughout the year, and throughout the last 2 years, to see how they've been surviving on the seabed. And we were lucky enough to use a boxfish, which is sort of an underwater drone, to film one of the mussel beds. And we made this 3D model to show people what these kind of small plots look like. And this is a plot from Grant Bay, that inner Pelorus Sound location. And this location has a large amount of very large starfish, which you can see here kind of mulling over the bed. And because this is a rocky site, the mussels are kind of bumpy and have different sort of ridges and habitat complexity. And other scientists have shown that that habitat complexity can really help with survival and things like that. And so this is a graph, to show you guys the survival over 2 years we monitored these beds. So here is the x-axis, and this is time in months, so 24 months, 2 years. And this is a y-axis to show survival. And then each of these colors and lines represents one of those 5 locations. So 4 of our locations have done really well after 2 years. And those locations are the ones in the Kinapuru and one up the reach, Maori Bay. And so they have kind of varying survival, but the lowest is just above 80%. So they're still doing really well after 2 years. And then you can see in this dark purple color in these circles here that we had one location completely die after 18 months, and that was Grant Bay, that inner Pelorus site that was rocky that I showed you from the model. And what happened there was we went to monitor the mussels at this after 18 months. And here we have a picture to show you of what that looked like. So we had a lot of dead mussels, which you can see here from sort of the open shells. And we had the starfish eating them still. So we had a large amount of starfish at this location over time. And I'll show you that on the next slide. And then, yeah, we still have some thriving beds after 2 years. So the good news is we did find locations where the benthic environment is suitable for mussel survival, which is great. And we also learned that sort of that mid-Pelorus area has a lot more starfish, and the starfish are eating the mussels, and they're not only more starfish, they're also bigger. So understanding the predator-prey sort of dynamics is a really interesting for future work. So here we can show you how many starfish were on each of these beds over time at each of these locations. And so here's a graph showing you each location and how many starfish we actually physically counted, but we also picked up and removed and relocated these starfish at each sampling point. So each of these times, the starfish had actually re-invaded the mussel beds. And so in our Kinniperu sites, Skada, Tamara, and Weka, there's low starfish over time. And then going sort of out of the Kinniperu and up into the inner Pelorus, we're seeing more starfish, but particularly that Grant Bay site had experienced quite a lot of starfish. And if you see here, after 5 months on the seabed, we collected 140 starfish from these small plots. And here is a photo to show you sort of what that looked like. So we had very large starfish. And then when we collected them, they actually created a bag that was 1 meter tall. So just basically, we've attributed the death at that location to starfish predation. But understanding these starfish dynamics and how many and what size are at these different locations has led us into understanding suitable habitats for restoration in Pelorus Sound in the future. And if we are going to continue to try to restore in that mid-Pelorus area, we're going to have to try to mitigate some of these issues, some of the starfish issues, or protect the mussels from starfish in some way. So we had also done a biodiversity assessment on these mussel beds. So this is an assessment to look for what kind of organisms are living inside the mussel bed, which we refer to as our epifauna. So those are things like crabs and starfish and different types of algae. And then we also wanted to look for what types of organisms are living underneath the mussel bed, so inside the sediment, and how the mussels sort of impact the sediment. And then what type of fish and different pelagic organisms are visiting the bed or living inside the bed. And so we did a full assessment at 6 months and 1 year. And we did that by taking video transects, so taking a video camera and recording the mussel bed while diving. And then we took these cores to capture all of the organisms in the sediment. And then we put these cameras down to record the fish populations that were visiting and living in the beds. So here's some videos from that to share. So we saw a lot of triple fins. So that was a little triple fin living in the beds and being very territorial, actually. And we saw those as soon as 5 months. And we also saw these blue cod. So we saw lots of blue cod in one of our sites in the mid-Pelorus, the Maori Bay site. And we saw them sort of in the beds, visiting the beds, hunting in the beds for food, more often than on control sites. And we also saw some bigger pelagic organisms like stingrays. And in the North Island, stingrays or eagle rays have been shown to eat the mussels, but we didn't see any of that happening here. It's really interesting. And then we did see, this is an off the plot sort of camera as a control, but we did see some very cute seahorses, and we have seen them in the plots while we've been scuba diving. So just to give you guys an idea of some of the organisms that we've been seeing. So this is a summary of what we saw in our biodiversity assessment. So we saw 4 times more blue cod on the mussel beds than in control areas. We saw 66 times more triple fins on the mussel beds. So triple fins are building their homes in these mussel beds. And like I said, we saw this each time we visited them via scuba, but we saw this in our biodiversity assessment where at 5 months and at 1 year. And we saw 23 times more sea cucumbers, which is very cool because sea cucumbers are a pretty cool organism. And we saw 2 times the macroalgae, so different types of algae living on the mussel bed. So this is just a really good finding because we know that wild mussel beds and different other shellfish habitats kind of create biodiversity and create habitats, but we didn't know how quickly restored mussels can build these types of habitats. And so we know now that this happens very rapidly after putting them on the seafloor. So as soon as 5 months, and they will build this type of biodiversity. And we saw this on these small plots that we put down. And so restoring mussel beds on a bigger scale will restore these ecosystem services that they have as well. So very interesting. And next, we tested the use of recycled mussel shell from industry. So we took these half shells of mussels that would normally go to a landfill, and we put them on the seabed to see if we created a substrate, if that would enhance mussel survival or these biodiversity and habitats, things like that. And after 1 year, here we have our mussel survival. So we have our shell plots in 2 locations, Fairy Bay and Kinniperu entrance. And we have our mud plots, which are just our control plots, mussels on mud, in also Kinniperu entrance and Fairy Bay. So this is a different experiment that we used in 2 locations. And what we found was by putting the shell down, it actually didn't seem to make a difference after 1 year in mussel survival. So our first experiment told us sort of where mussels can survive and what types of impacts they're going to be having on the environment, but also what type of predation and starfish are we going to see there. And then we used that information to sort of apply to the second experiment using recycled shell to see if we could use the shell to restore mussels. And yeah, and what we found is the shell didn't make a difference to mussel establishment. The mussels can establish either way. But some pretty cool benefits that we did see from the shell is that if you see here, the shell layer also has some biodiversity. So it has different organisms that kind of move in and use it as a habitat. And so we want to, in the future, explore this shell substrate further and how we can use just the shell to help restore some of the benthic environments and potentially create a habitat for new juvenile mussel recruitment. So very cool. And the third experiment that we have is this experiment here, where what we did was we wanted to test mussels in the intertidal area. So that is exposed at low tide on the shoreline where people used to hand pick mussels. And we wanted to put mussels down to see if they could survive in this sort of more harsh environment. So where they used to thrive. And so what we did was we put mussels in intertidal and subtidal as a comparison. And then we wanted to test two different size mussels. So we put down subadult mussels and adult mussels to see if we can use different size mussels for restoration and in these two different depths. So that experiment went down onto the seabed in June 2021. So for the time I'm recording this, a year. And the results of that are still coming in, but what we have seen is in that intertidal zone where the mussels are exposed at low tide, it did get quite hot over the summer, and the summer heat did affect the mussels depending on how high up the shoreline they were exposed. So the less exposure time, the better for survival. And we didn't see any differences between using subadult mussels, so those smaller mussels compared to adult mussels, which is great news because that means we can use different size mussels for restoration, and we can use those faster because they don't have to grow all the way to the adult size for us to use them for restoration. So really cool, and we're still monitoring these mussel beds. So for this restoration project, we have the portion that I've sort of talked about and is coming to an end, and we're trying to explore options to continue this work, super important work to restore mussels in Pelorus. But also we have recommendations for the next steps for this project. So unfortunately, we didn't see any juvenile recruitment into the mussel beds for any of our experiments. And some of them over 2 years, some of them over 1 year, and we just didn't see any new juvenile recruitment. And so we think that there might be some sort of bottleneck where the new recruits, so the new baby mussels, are having a hard time getting into the mussel beds. And so we want to explore that further, because real long-term restoration success would be to have new juveniles recruiting into these restored beds to help replace the ones that sort of naturally die over time. And that will ensure that these mussel beds will remain long-term. And we also want to restore this recycled shell as this benthic layer further, and kind of understand if the recycled shell can be used to bind sediment and build habitat in a way that would be able to cover larger amounts of area faster, because these shells are a byproduct of the aquaculture mussels. So we can use these to help restore some of the environment. And then we would like to move into different areas in the top of the South Island and use these methods and the harvesting methods and the deployment methods that we have learned and trialed in this project to expand those to be able to work in different areas, including Golden Bay, which is a more exposed area than the protected Pelorus Sound, and sort of try these different methods and be able to use them elsewhere throughout New Zealand. So those are sort of our next steps for this project.
We really enjoyed working on this project, and it was great to be recognized at the 2021 Cawthron Marlborough Environment Awards, where we won the marine category, which was sponsored by Port Marlborough. The awards judges said the project works on a number of levels. It is restoring the marine environment and engaging with marine farmers, and there's also potential where there's community involvement. They also said that this project sets a great example for industry groups to take responsibility for the environment that they operate in. The judges' report was impressed with our scientific approach, strong collaboration, and the potential to scale up the project as research and monitoring results build up. Entering the Marlborough Environment Awards was a really great experience for us, and we really appreciated the recognition.
So I just want to take the time to thank the many, 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 many hands that have made this work possible. We have our team of scientists, which includes Dr. Andrew Jess, Dr. Jen Hillman, Dr. Sean Hanley from NIWA, and Trevin Toon, my colleague from NIWA as well, and Ned Wells from the Marine Farming Association, and just all of the marine farming companies that made this work possible. I cannot thank you all enough for just the extensive support, the knowledge, the encouragement, every step of the way in this project. Thank you all so much. And if any of you have any questions, you can reach out to me personally via email, or you can check out Twitter, which I share a lot of information about this project. And also we're on the Marine Farming Association website, and there's different avenues that you could find more about our project, but feel free to reach out if you have any questions or anything at all. Thank you so much, and thanks again for listening to this talk.